started. Hi, I'm Jen, and I'm going to talk to you about the evolution of hybrid seed viability in the world's best genus, Mailis. So I'm sure I don't have to convince anyone in the room that the world is a pretty speciose place, and as evolutionary biologists, we're quite interested in trying to understand how this type of diversity is first formed and then maintained through time. And essential in both the formation and the maintenance of species is the formation of barriers to reproduction. One very important barrier to reproduction in seed plants is hybrid seed viability. So here's some data that's just been lifted from Dave Lowery's paper, and you can see that hybrid seed viability is fairly common and also fairly strong in a lot of different diploid plant species. Now, despite its commonality, we actually don't have a very good understanding for why a lot of intrinsic postzygotic barriers evolved to begin with. One potential hypothesis is that of parental conflict, which is just simply the idea that moms and dads don't have the same optima for resource allocation to their kiddos. That's because moms are equally related to all of the offspring that are produced, and so selection should favor equivalent resource distribution among them. However, in any system, or in systems where there's any amount of non-monogamous mating, dads are not equally related to all of the offspring produced, and so selection can favor the evolution of greedy alleles. This tension between moms and dads can result in a coevolutionary arms race, where dads are selecting for greedier and greedier alleles, and moms are selecting for more repressive alleles. Now you can imagine this might manifest as reproductive isolation of two populations that have been evolving at different rates or in different trajectory in this coevolutionary arms race, come back them together in secondary contact, and they're no longer a match for these maternal repressive or paternal greedy alleles. Now this is particularly true in a couple of systems, placental mammals and seed plants, where there is a special nutritive tissue that is a direct nutritive connection between mom and offspring. In seed plants, this is the endosperm, which is formed from a fertilization event between pollen grains and this thing called the central cell. So that makes this endosperm that is a special genetic composition of 2 parts mom to 1 part dad. And it turns out that ratio is very important for proper seed development. We know from interploidy crosses, where you're manipulating either the maternal dosage or paternal dosage, that we get these endosperm phenotypes that are very reminiscent of conflict, either overgrowth or undergrowth. And it turns out that 2-to-1 ratio is very important because many genes that are expressed in the endosperm are imprinted, meaning they have non-random expression depending on whether they're maternally inherited or paternally inherited. But I told you that most hybrid seed viability, or a lot of hybrid seed viability we see in nature, is actually between diploid species. And so the questions then remain, is it that these genes that are involved that are imprinted in the endosperm and important for endosperm development are the ones that are causing hybrid seed viability? And are they evolving through parental conflict? So I'm using Mimulus to try and understand these kinds of ideas. Mimulus is a great system for this because hybrid seed viability has evolved multiple independent times throughout the genus. Today I'm just going to focus down here on the Guttatus species complex, which is perhaps a species complex many of you have heard about. They're well known for these extreme and beautiful adaptations in the wild, and they're a great system for understanding differences between life history types. However, in this complex, there's also a lot of these perennial morphological variants that have been relatively ignored. And today I'm going to talk to you about this thing called Decorus, which looks a lot like perennial Guttatus, except it has a slightly more hairy stem. You can imagine why this was ignored for a long time. I'm also going to mention Mimulus Guttatus, its closest sister species, which is comprised of a bunch of different life history types. I'm just going to all call Guttatus. And then I'll probably mention this thing called Telingia, which is a very divergent perennial species. Okay, so to understand this Decorus thing, we went out to the field. We collected a bunch of different samples of Decorus throughout its known range in Oregon and Washington. We whole genome sequenced those, as well as a bunch of different genomes of Guttatus, and then some others from the complex that I won't really talk about today. We also completed a common garden study and measured a bunch of different phenotypes there. So let's just hop right into the genomics. So what you can see down here is that all of our Guttatus samples, which are shown in pink, are a big cloud down there. So regardless of life history, they all cluster together. But our Decorus is that weird spread way up in the left-hand corner. And so it's quite divergent from Guttatus in this PC space. And you'll see that I've colored it in 3 different ways. So in the process of this study, we found that Decorus is actually this polyploid complex. And we found a bunch of tetraploids, which are shown in green. And I'm not going to talk about them much, but I'm happy to talk about them later. And we also have these 2 diploid clades, which I'll show in yellow and blue. Okay, So moving on to the phenotypes next, we see that our 3 genetic clades of decorus are actually not phenotypically distinguishable from each other. They're shown here. They're stacked right on top of each other, and they're all stacked right on top of Bradyocutatus. So just as I mentioned before, these are a bunch of species that really don't show a lot of phenotypic divergence, but they're genetically quite distinct. As speciation biologists, we know that the currency of speciation is reproductive isolation. So we next took actually 21 populations, not 19, and we crossed them to 4 populations of Cutatus reciprocally. Again, I'm just going to focus on the diploids for now. Happy to talk about the tetraploids later for anyone who wants to know. And what we notice when we cross these 2 species is that in one direction of the cross, the seeds are nice, plump, and viable. Those will germinate. In the other direction of the cross, they're inviable, shriveled, sickled things that are just never going to germinate. But what was curious to us is that our 2 diploid genetic clades of decorus actually showed oppositely asymmetric reproductive isolation with Cutatus. It's important to note that our populations of Cutatus did not show any hybrid seed viability within them. And so we wanted to leverage this diversity that we see in hybrid seed viability to see if we can't learn something about conflict. So we made 3 predictions. The first is that if we have these parent of origin effects on growth, those reciprocal F1s should look different. They should be different sizes. They're just simply measured seeds. We see that in both sets of our crosses, reciprocal F1s look quite different. Okay, so that's great. We would next predict that if hybrid seed viability is caused by these parent of origin effect alleles, that the extent of difference in size between those reciprocal F1s should be correlated with the extent of hybrid seed viability. And in fact, we see just that. So this is just a relativized measurement of the differences in reciprocal F1s and their size and their ability to germinate, where 0 means they're the same. So we see the strong correlation. And lastly, we would predict that if we can infer the history of conflict within a population, that that history of conflict should be pretty static. It's not going to be context dependent. That means that if we have a relatively strong population and a population that experienced relatively weak conflict, we should be able to predict the outcomes of crosses we've never tried before. So in order to do this, we have to make some inferences about the history of conflict in our populations. And to do that, we're going to go back to our seed sizes from our crosses with Cutatus. And we can think of Cutatus like a tester cross that is telling us about the history of conflict in this group. So what we actually think is happening is that our northern decorus has a relatively weak history of conflict, or history of weak conflict. And that's because when they're the maternal donor in these crosses, those seeds are overly large, which means they're unable to repress those greedy Cutatus paternal alleles. In contrast, our southern decorus actually shows the opposite pattern, which means that those Cutatus maternal alleles are unable to repress those greedy southern decorus paternal alleles. So what we think we actually have is a situation in which our 2 clades of decorus exhibit the most extreme conflict phenotypes, with Cutatus somewhere in the middle. And I've just put our tetraploids back up there because just by nature of doubling the genome, you're doubling expression of those maternal or paternal genes. And so we can make the prediction that anything that's far away from each other on this continuum should have strong reproductive isolation. Things that are closer together should have much less hybrid seed viability. Or simply put, those northerns crossed to either the southerns or the tetraploids should show a lot of hybrid seed viability, whereas tetraploids crossed to southerns should show relatively minor hybrid seed viability. So to do this, we took a focal population from the north and one from the south, and we just crossed them to everybody. And what we find, if you take that focal population from the north, you cross it to all the southerns, the focal population from the south, and you cross it to all the northerns, there's 100% hybrid seed viability in both directions of this cross. And that's paired with reciprocal F1 seed size differences in the direction we expect from conflict. If you take that focal northern population, you cross it to tetraploids, again, 100% hybrid seed viability, again, reciprocal F1 seed size differences in the direction we expect from conflict. But if you take that population, focal population from the south, and you cross it to tetraploids, there's no hybrid seed viability, there's no reciprocal F1 seed size differences. So we next wanted to actually map hybrid seed viability. For this, we're just going to use our northern decorus population. And what we're trying to find is nuclear alleles with parent of origin effects. That would indicate that there's some sort of imprinting going on, which is consistent with conflict. So in order to do this, we took an F1, we back crossed it in both directions to both parents, And this gives us 4 different cross directions, which actually sets up a really nice comparison for us. And that's because this barrier is asymmetric, it means that when we use our F1 as either the maternal or paternal contributor, we are manipulating whether those alleles are inherited maternally or paternally. So this direction of the cross can tell us these gutatus paternal alleles, which are interacting with our decorous maternal alleles to cause a dead seed, whereas this direction of the cross, where you don't expect to see very much hybrid seed inviability or very much segregation distortions, we're using this approach called segregation distortion. And that just simply means that we take these seeds, we're going to plant them up. Of course, only the survivors are actually going to germinate and be sequenced, which means that the survivors are going to have a deficit of alleles that contribute to hybrid seed inviability. So we can just march along the genome, and we can look for regions of the genome that are distorted in one direction of the cross, but not in the other. So when we do this for this first direction of the cross, where we're looking for these gutatus paternal alleles, we find 5 regions of the genome which show strong segregation distortion in one direction of the cross, but no segregation distortion in the other direction of the cross. We can calculate selection against inheriting that gutatus allele paternally. You can see those selection coefficients are really quite strong. So these are alleles that kill you if they're paternally inherited, but you're totally fine if they're maternally inherited. We can do the same for our other direction of the cross, where we're looking for our decorous maternal alleles and some compatibility. And we see that there are 3 regions of the genome that show this parent of origin specific segregation distortion. Of course, though, maybe this is, again, such a great system for this. I'm not the first person to start studying this. In fact, many other people have been looking into this and starting to map things, including Andrea Zweigart at the University of Georgia and Elin O'Neill at Duke. And it turns out that they've also made QTL maps. So what I'm going to show you next is incredibly speculative, just to finish off my talk, and I'm happy to talk about it more. There's going to be a lot more work going into this, so don't get too antsy, but I'm happy to talk about it more. And that's just simply that I took the QTL map that Andrea made from her paper, and I just put boxes around the QTLs that either I found or Elin's found or all 3 of us have found. And what I want you to take home from this is that there are a lot of QTLs with boxes here, and actually of the 8 QTL that I showed you, 7 of them have been found in another cross. So just to sum up, what I've shown you is that there's a lot of cryptic diversity in perennials within this group. There's also a lot of hybrid seed inviability within the genus more broadly. Patterns of hybrid seed inviability are consistent with parental conflict. I've also shown you that the genetic architecture of hybrid seed inviability is quite complicated, involves these alleles with parent of origin effects. And there is potential, although I'm not taking any bets at it yet, for this parallelism among different incidences of hybrid seed inviability. So with that, I want to thank you all for coming, thank my collaborators, and particularly the undergrads who helped with this work for shown in red. Thanks.
You looked at traits for decorous and maybe gutatus and overlap. Is there overlap in the environment that they're found in, or anything climatic? Is that what it's found?
Yeah, so decorous is pretty much restricted to the Cascade Mountains in Oregon and Washington, as far as we know. And gutatus grows everywhere around there. So we haven't really looked, we haven't done any of this climate mapping. So there might be something about it.